Hey guys, and welcome back to this course. In this lesson we'll be studying about plasma membrane. This course is presented by me, Navneet. You can know more about me from here. You can follow me on this link, and if you like the lesson, kindly rate, review and recommend. Beginning with the lesson, the cytoplasm of the animal cell is bounded by a thin limiting membrane called as the cell membrane. Nageli and Kramer called this membrane as the cell membrane in 1855. In 1931, Plowe named this as plasma lemma. Modern cell biologists prefer to call it plasma membrane according to De Robertis. Danieli and Davison in 1952 proposed a molecular model of plasma membrane. Robertson after that proposed a unit membrane concept in 1960s. Okay, so these were the models just to explain the structure of a plasma membrane. The material used for study of plasma membrane, the human RBCs are kept under a dilute solution which is hypotonic, then it swells due to the intake of water. It breaks and protoplasm will go out. This is called as a hemolysis. The remaining membrane is called the ghost. It is used for plasma membrane studies. Liver cell or membrane surrounding the nerve fiber are used as material for the study of plasma membrane. Physical nature of plasma membrane. They nearly described the nature of plasma membrane. Robertson, by using electron microscope and X-ray diffraction methods, proposed the structure of plasma membrane. The plasma membrane shows three layers, that is, it is a trilaminar. First, outer layer is made by proteins, which is approximately 20 angstrom in length. Middle layer is bimolecular lipid layer, which is of 35 angstrom. Inner layer is made by a protein, which is of 20 angstrom again. Membrane model. The membrane models are many. These models will represents the molecular organization of the membrane. How the membrane is made, how and what particles the membrane is made up of. Danieli-Davison model. Okay, it was the first important model that we need to study. It shows a thin lipid layer with protein absorbed on both the sides. The lipid the lipid contains polar heads pointing outsides and ion polar parts run transversely. In 1954, Danieli modified this model and gave a new model. Okay, so you can have a look at the diagram. This is the Danieli-Davison's model of the plasma membrane. Okay. Next we have the Robertson unit membrane concept. Robertson in 1959 described the trilaminar structure of plasma membrane again. He modified it as it contained an outer and inner protein layer. Okay, in between them a bimolecular lipid layer is present. Okay, so there is an outer and inner protein layer, and a double molecule, that is bimolecular lipid layer, is also present in between them. The unit membrane is 75 angstrom thick. The outer and inner protein layers are 20 angstrom thick. Okay. In between them, lipid layer shows 35 angstrom units. Okay. So this is it. The polar hydrophilic ends of the lipid layer will face the proteins, while the hydrophobic ends of the lipids are away from the proteins. Okay, so this is the Robertson's model. So you can have a look at the diagram. This is the outer layer, this is the inner layer, and that is the lipid bilayer. Okay. Next we have the most important fluid mosaic model of the plasma membrane. In 1972, Singer and Nicholson were the two scientists who proposed this model. Cell membrane, according to which a cell membrane is a mosaic of lipid and proteins. Lipids are arranged in a bilayer way. It forms the structural framework of plasma membrane. Protein molecules are arranged in two ways. Okay, firstly they can be arranged like the extrinsic proteins. These proteins are located adjacent to the outer and inner surfaces of the bilipid layer. Secondly, we have the intrinsic proteins. These proteins will penetrate into the lipid layers partially or totally. They are called the integral or intrinsic proteins. This fluid mosaic model is accepted universally. Okay, so once again we shall have a look at the diagram. So this is the fluid mosaic model. We have the transport proteins. These are the intrinsic proteins, and the peripheral proteins are found on the outer parts. And this is a lipid bilayer. Okay, and the carbohydrate chains protrudes out from the protein chains. Okay, so it is clear from the diagram. I hope so. Next, we have the chemical composition of the plasma membrane. The plasma membrane shows proteins, lipids, and small percentage of carbohydrates. Firstly, we have the lipids. The lipids of plasma membrane are phospholipid, lecithin, cholesterol, glycolipids. The distribution of the phospholipids in the bilayer of plasma membrane is highly asymmetrical. The phospholipid will show two ends. First is hydrophobic end, the second is hydrophilic end. Okay, hydrophobic, clear from the name itself, means it is a water hating, means which has a phobia from water. Okay, so it is a water hating non-polar end. And the hydrophilic end means the end which loves water. It is the water loving part, it is called as the polar end. It is present near the proteins. The hydrophobic end contains fatty acids and hydrophilic end contains the phosphate group. That's why they have this kind of property. Okay. Next we have the proteins. The peripheral proteins or extrinsic proteins are free from lipids. They are loosely attached to the membrane. The integral proteins or intrinsic proteins will penetrate into the lipid. These proteins will give mechanical strength to the plasma membrane. They also regulate cell activities by facilitating the transport. Okay. Next we have carbohydrates. A very small percentage of carbohydrates are present. Hexoses, hexosamine, fructose, etc. are present. They may be attached to the lipids, glycolipids, and they are attached to the proteins as glycoproteins. These glycolipids and glycoproteins are present on the external surface. Okay, so they attach with the lipids and form the glycolipids, and they attach with the proteins and form the glycoproteins. Okay, so next we have the enzymes. So more than 30 enzymes are isolated from plasma membrane. They are phosphatase, ATPase, RNase, etc. Okay, ATPase means like ATP degrading enzyme, RNase means RNA degrading enzyme, phosphatase means phosphate degrading enzymes. Okay, pores in the plasma membrane. Plasma membrane shows some pores. The diameter of them is approximately 35 nanometer. Sorry. The modifications of plasma membrane. To perform the specialized functions, plasma membrane will show some modifications. They are microvilli, interdigitations, desmosomes, tight junctions, etc. First we have the microvilli. In certain parts the plasma membrane will show minute enfoldings. They are called as microvilli. They show 1 millimicron diameter and 6 millimicron length. In some cases, the villi are connected with the endoplasmic reticulum. These microvilli are more in intestinal mucosa cell. The microvilli shows cytoplasm with microfilaments. The microvilli will increase the absorptive surface of the intestinal epithelium. Okay. Interdigitations. At some places, the plasma membrane of the adjacent cells will develop into the finger-like projections. They are called as interdigitations. The desmosomes. The plasma membrane of adjacent cells become thicker in certain regions. On these thick areas, fine filaments are present. They are called as tonofilaments or tonofibrils. Such parts are called as desmosomes. So the desmosome is concerned with the cell adhesion and maintenance of the cell shape. Terminal bar. It is a desmosome without tonofibrils. It is also called as the terminal bar. Zona occludens. So these are the spatial area of adjacent cells where the two plasma membranes fuse. Okay, so these tight junctions are present below the apical border. It gives a mechanical attachment between cells. Okay, lastly we have the properties of plasma membrane. The substances which either enter the cell or leave the cell should pass through the plasma membrane. Okay, the plasma membrane will show permeability. It shows the following properties as it is selectively permeable. Okay, osmosis, passive transport, endocytosis, pinocytosis or cell drinking, micropinocytosis, phagocytosis, exocytosis or active transport. Next, we have the origin of plasma membrane. It is believed that it developed de novo. It is formed by the assembly of lipids and proteins. Okay, so this lesson ends here. I hope you guys like the lesson. Thank you guys for watching.